Hello, and welcome to our continuing Texas 4000 speaker series. This is an opportunity to hear firsthand from doctors and cancer experts about a variety of topics. I am Ryan Logan, the 2021 Programs Chair, and today we are joined by Dr. Bill Matsui from Dell Medical School to hear about some topics in cancer research and hematological malignancies. Dr. Matsui is the Deputy Director of the Livestrong Cancer Institute and a professor in the Department of Oncology. Today, we're going to talk about some of the research he's been doing and how Texas 4000 has been able to help with that. So whenever you're ready, Dr. Matsui.
Sure. I thought, first off, I want to thank the Texas 4000 for all of their support. And, there was a very specific project that we wanted to work on with the help of Texas 4000. So this is really the start of it. And so I wanted to give some ideas about sort of what the idea is and sort of where we're going with this. So if I can have the next slide. So one issue is that we know that there are racial differences that exist in cancer and cancer care. These can range from like genetic differences within tumors all the way to, things about healthcare and healthcare delivery. So the one thing that we're interested in is, are tumors, the same type of tumors, different when they arise from different racial backgrounds? So we know these exist in a number of cancers, but what their implications are, what the causes are unknown. The disease that I study the most is called multiple myeloma, which is a cancer of the immune system. And patients who are African American have a higher incidence of the disease and have worse outcomes. And this is especially the case for African American males. So our ideas are to, if we can find and understand genetic differences within the tumors, then maybe that leads to more specialized treatment strategies that will, erase these differences between racial groups. So if I could have the next slide. So this is a graph of just looking at different mutations. So across the bottom are a bunch of different genes. And these are from one study called the COMPASS study, which looked at gene sequencing of a bunch of different myeloma samples. And the way that they were able in this study to figure out racial background is by looking at the same sort of things you look at in 23andMe. So you can sort of genotype people that way. So if you look at, certain genes, they are much more frequently mutated in African-American cases of multiple myeloma than in those of patients of European descent. And so there's just a list here of, these are the ones that really stand out, but there are a number that are higher in African Americans and there are a couple that are lower. So we know that there's some differences that exist. So in the next slide, what we're looking at is just sort of, is there an implication for these differences? So on the left, it's what's called a survival curve. So at the bottom, you have a duration of time. And then on the y-axis, you have the proportion of patients that remain alive. So everyone starts out alive, they're 100%, and then over time, patients will die. And so if we look at all myeloma cases and we go by, and this is by self-reporting, and we look at race as a variable, you can see that the line in blue is for people who identify as white. And below that in the green line is those who identify as Black and African American. And so there is a lower number of patients or a lower curve, and that means that those patients are dying at a faster rate. So if we go in and we now look at BCL7, which is one specific gene that is overexpressed or more frequently mutated in the disease, here what we can find is that this also causes a separation where the green line is lower than the blue line, so patients are dying more rapidly when this gene is mutated. So in the next slide, the question is that, so how do we sort of now study this? Because that's all sort of population-based analysis. So what we do is we're trying to develop models where we can actually change the amounts and the way that BCL7 looks. So for many gene mutations, they're within something called the coding sequence, which is like the gene itself that makes a protein. And so you can have differences there that alter the amino acid sequence, and that can cause a mutation. The other way you can do it is something where they're in the non-coding regions, and what that does is generally affect the level. So in patients with these mutations, it's not that the gene itself is altered, like the protein it's made is not changed. It's just that the levels of it are higher or lower. And so what we have here is a model system in which we can manipulate very easily the amount of BCL7. You can see that sort of squiggly line up at the top. And we can manipulate it so that we can have a little bit of it, a lot of it, or it goes away. So when we do that, if you look at the bottom two panels, what we do is we take cells that either have, regular BCL7 levels or those where we've like knocked out BCL7 levels, and we look at their ability to form tumors. And so if you lose BCL7, then you can form tumors at a more a much higher rate. And so what that suggests is that there are some biologic changes because of BCL7. And so this gives us like a toolbox that we can now manipulate and further study. So really the next step right now is to sort of understand like how do these mutations, cause a higher frequency of disease, and how does it regulate specifically a progression event. And what I mean by that is that in multiple myeloma, a lot of people will have a precursor condition called MGUS. And so that's where you can see sort of abnormal cells, but they're not at a high enough level that causes any health problems. And so when you get those health problems, that's when you turn into an actual myeloma patient. So in African-American males, they have a much higher incidence of this precursor condition. And because of that, they transition over to myeloma at a higher rate. And so what we want to do is we want to sort of study how these mutations, do they play a role in that aspect of the disease? And the reason why we want to do that is that, normally if you have MGUS, this precursor condition, it's like you transition to the cancer at a rate of 1% per year. So not everyone for sure is going to get it. Some people will, but it's thought that if you treat these conditions at the earliest point, it's sort of like if you get a colonoscopy, there's a polyp, you take it out, that's not going to become cancer. So if we can do that with MGUS, then maybe what we can do is prevent cases of myeloma from actually happening. And then the other is we're thinking about doing something where we look at, all of the things that interact with BCL7 to try and figure out, well, what are things that, if you're missing BCL7, does it provide some unique susceptibility, right? So there's some pathway that they're now dependent on. There's some factor that is also altered. So if we target that, like it's hard for us to target something that's missing, but whatever the downstream consequences of that are, we can figure that out, and then we can try to find new ways of dealing with these cancers. So that's the next steps is what we're gonna do with the funding from Texas 4000, and that's I wanted to give you a background of where we sort of came up with the idea and what we have so far.
Thank you so much for that presentation, Dr. Matsui. It was truly insightful. All of us on Texas 4000 are so happy to be able to contribute to your work in any way we can. Now we will transition to a quick Q&A session with some other members of our programs team.
Hi, Dr. Matsui. I'm Yash. I'm a member of the Ozarks 2021 team. I just want to start off by thanking you for that presentation again. It was, again, extremely insightful, and I got a lot of great information out of it, so thank you for that. My first question is in regards to clinical trials. So what is the basic process of science research as it pertains to clinical trials?
Yeah, so basically, most ideas start at a basic level, right? So if you think about a lot of sort of great things and vice versa, so a lot of great sort of findings in basic biology have come from studying diseases, right? It's like sickle cell, like you understand how proteins start to work. And so the process by getting a basic science idea into a clinical trial is you have to go through something called a translational research time. So basically you take that idea, it's the easiest way to do it is to take, things and make drugs that do something, right? And so if you can now go through the process of understanding a biologic process, finding chemicals or drugs that can interfere with that process, and then you want to make sure it works, you want to figure out what diseases it works in, and then with that you try to go into the clinic with, let's say, that drug. So that is a process where you might say, oh my God, that seems like such a random process. Like, how is it ever going to work? And so most of the time it fails. But I think that once you get certain basic ideas into the clinic, then you can really build on them. You combine drugs, you find other ways of inhibiting, so you can really enrich the what we do with patients. So one really great example is something called proteasome inhibition, and that's an idea that's been around forever. It's a way of cells to get rid of excess proteins. And in myeloma, they make a lot of proteins. So if you block their ability to degrade it, the cells totally get full of protein and freak out and die. So that is really one of the main ways we treat myeloma patients these days. And that all came from a very basic idea. And the people who came up with that were much smarter than I am, but it's really like a pillar of what we do clinically now. So there are a lot of amazing stories about what we do in the clinic now, which for sure didn't exist 20 years ago when I started practicing. And they all are, like, the vast majority come from some sort of bizarre basic science place.
Awesome. Thank you.
Hi, Dr. Matsui. I'm Addison, and I'm also on the 2021 Ozarks team. Again, thank you for your presentation. It was very good, and I got a lot of information out of it. What should people know or ask about before enrolling in oncologic clinical trials?
So there are a bunch of different kinds of trials. So there are trials that are like where you just get followed, like there's no intervention, right? So it's an observational trial. There's usually no harm that comes from that, right? So there's no therapeutic thing. There could be trials that now test new drugs or compare different combinations of drugs. So those are therapeutic trials. So those are obviously at a different level. And so what you need, I think, if I were in the position, what I would want to know is that what would standardly be done right now with me? Like, what would I normally get? And then in a clinical trial, like, how is that supposed to advance that? Like, is it something new? Is it something where we think something is better, but we don't know yet, right? And then the third thing I will say is that the one thing we don't do a lot of in oncology clinical trials, we don't do a lot of placebo-controlled trials, right? So if you had cancer and you say, okay, well, I'm going to go into, I heard about this trial, you either get the cancer treatment or you get, like, Tic Tacs, I would sort of say, well, who would be crazy enough to go onto that trial? So I think that you have to sort of understand what's the standard, what all are the alternatives, and what exactly are they testing in that trial? Like, every trial should have a specific question it's trying to answer. So if you understand that and it makes sense to you, I think a trial is a great way to go about it. The one thing I will say about clinical trials is that they're really, really good for patient care because everything is laid out very strictly. When you come in, what labs you get, when you get seen, all of this stuff. And so it is like the way to get the best care possible is to do it on a trial. And so that's one of the major benefits of doing things on a clinical trial.
Thank you for answering that.
In that same vein, what disparities exist in clinical trial enrollment and how can they be addressed?
So this is like, if you could solve this problem, this would be like one of the biggest things in oncology. So there are differences in who enrolls in clinical trial. I think that, there are different forms of sort of trust in the medical system, right? And that goes across different populations. It goes across different geographical areas. And so I think that a lot of it, it does, not only just in terms of like taking care of people, it goes to clinical trials because, what we don't want is we don't want people to feel like they're a guinea pig for something, right? Like, that is not the case. We're trying, in this context, we're trying to help you and we're trying to help people who are going to be coming after you. Right. And so I think that needs to be understood. And I think a lot of it has to be a sense of education that clinical trials are not there to have just to test something on you. There is a perceived benefit to them. And I think that if it were, and this is a strategy, is that you have to go into places and talk to people who have influence over a population or a geographical area or something and convince them, and then they can spread the word. Like, an outsider coming into a community and saying, look, you got to go on clinical trials, that's probably going to prevent more people from going on clinical trials. So you really want to get in there with leaders in those communities. And this is not just for cancer, it's for like every sort of health condition. And so you got to understand the patients, right? And you got to understand how the patients perceive things. And then you got to provide like a reasonable, cogent argument to them why they should participate or not. And that's like the only way you're going to do it. You can't force someone to do something. You can't fool them into doing something. You have to get them to believe that this is the right thing for them.
Thank you. That was insightful.
I have a question regarding some of the information that you presented in your PowerPoint. Are there any environmental factors that might have some, say, epigenetic consequences which could possibly exacerbate or kickstart myeloma progression in those with MGUS? And if so, are perhaps African Americans more exposed to these environmental factors in comparison to the general population?
Yeah, so there are a lot of environmental factors that have been associated with cancer, like smoking is the biggest one, right? So in blood cancers, there are very, very few that have been environmental things that have been associated with the development of blood cancers. In terms of MGUS, there is like, one of the things that's interesting is like, so people who are like first responders who were in 9/11, right? For whatever reason, they're getting a lot of MGUS. So it's not clear why that is. It must have been some sort of exposure. So that we're trying to figure that out. The other is that like people who were in, the Vietnam War who got exposed to Agent Orange for some reason had a much higher incidence of MGUS. And so it makes sense, right? You got to be exposed to something. We're just not smart enough to figure out what it is. And so that is like, a great thought that, could it be epigenetic? Because when we compare, like, there are these specific mutations, but you find them in MGUS and you find them in the myeloma. You just find them more frequently in one racial group or another. But there are not a lot of genetic changes. So epigenetic changes may be the key of it. And, we're getting better and better at studying epigenetics. And so that's something where, like, even at the Livestrong, people are interested in, like, how there are modifications to DNA and does that matter in cancer? So there's a big interest in that, across the country in terms of thinking about cancer development.
Thank you.
Texas 4000 partners with Be the Match, which is a national bone marrow registry, kind of similar to clinical trials and enrollment. Can you speak about the importance of enrollment in bone marrow registries and the power of blood and bone marrow stem cell donation?
Yeah, so one of the things that's happened in bone marrow transplantation is that it's much safer to get a transplant. Like, we use lower levels of chemo. And so when I started out many years ago, the oldest person we would transplant is someone like maybe around the age of 55 or 60, okay? But myeloma is a condition where the average age is in the 70s when you get it, right? AML, acute myeloid leukemia, is the average age is like 68, right? So it's well beyond the age when, we would have transplanted people. So most of the transplant patients were young. Nowadays, because it's safer, we can transplant people like in their 80s, right? Like, you might say, well, you're crazy to do that. But one of the interesting things that's been identified is that the younger, and normally what we do in a bone marrow transplant is that the ideal match is a sibling of yours that matches you, right? But if you're like 75, you're not going to have a 15-year-old sibling, right? You're going to have like a 70-year-old sibling. So one thing that people have identified is that the younger the donor, the better the outcomes are in a transplant. So if you have a 75-year-old, you don't want to use their 72-year-old sister, right? What you want is to find someone that matches who's like 25, because then their chances are much better. So really having this pool of donors is super important because it allows individuals who are older to find younger donors. And that, I think, makes a difference in the end. I think that like a lot of people like sort of sign up for these things, and then, I think it's about half the people who, when they're asked to donate, say, I don't want to do it, right? And so I think that one of the questions is, well, how do we change that? Is there some way of changing that? And I think that, there have been thoughts about being more educational about sort of, if you sign up, like what is what is it that you're going to do? Because time is of the essence when you're going to have a bone marrow transplant. So you can't go to a donor, track them down, find them, and then talk to them. And then like they say, well, let me think about it for a week. And then they say no. And then you got to find another donor and you got to go through this process. So by that time, if you do that long enough, the patient will have been dead of cancer, by that time. So I think that trying to decrease maybe the refusal rate is an educational thing, but they're going to play a much bigger role as time goes on, as people realize the younger you are as a donor, the better, the more you're going to help. So I think that this is an important thing, especially for you guys all being young. I'm sorry, you don't want my marrow. I want your marrow, right? Like that's the way it's going to go.
Thank you for answering that.
All right. Well, I think that just about wraps up all the questions we had. On behalf of all of Texas 4000, thank you so much for making the time for us and sharing all of that vital information. We wish you the best of luck moving forward.
Thank you so much for allowing me to talk, and thanks so much for your support.
Absolutely.